What if I told you we have a technology that can find a single misplaced letter in the 3 billion letter book of our DNA and fix it? It sounds like something straight out of science fiction, right? Well, it's not. It's a real tool called CRISPR. And today, we're going to unpack the incredible story of how this thing was discovered, how it works, and how it's already, literally, changing the world as we know it.
So let's just get right into it. We're talking about a full-blown genetic revolution. And it's a story that's got everything. A mystery that lasted for decades, a chance meeting that changed everything, and a technology so powerful it earned the highest honor in all of science.
And that big honor came in 2020. The Nobel Prize in Chemistry went to two incredible scientists, Emmanuelle Charpentier and Jennifer Doudna. And look, this wasn't just another award. This was the world saying, wow, okay, what you've done has fundamentally changed the life sciences forever.
The official words from the Nobel Committee really nail it. They won for the development of a method for genome editing. And that word, method, is so important. See, they didn't just discover some weird thing in bacteria. They figured out how to turn it into a programmable tool that any scientist anywhere could use.
And if you think this is all just happening in a lab somewhere, think again. Just in 2023, we saw a massive leap forward. The very first therapy based on CRISPR, it's called Casgevy, was officially approved to treat sickle cell disease. So yeah, we have officially moved from the lab into the real world.
But here's the thing that gets me. For most of us, CRISPR just kind of burst onto the scene around 2012, right? It felt like it came out of nowhere. So how did this overnight success actually take, get this, more than 30 years to unfold? To answer that, we've got to rewind way back, before the Nobel Prizes, before the big companies and headlines. We have to start with a little mystery that was hiding deep inside the DNA of microbes. And you know, the person who first tripped over this mystery wasn't at some famous, fancy lab. Our story really starts with a Spanish microbiologist named Francisco Mojica at the University of Alicante. He's really the unsung hero who got this whole ball rolling. And just look at this timeline. It shows you just how much persistence this took. All the way back in 1993, Mojica notices these weird repeating patterns in bacterial DNA. He just keeps chipping away at it, and in 2000, he gives it the name CRISPR. But the big eureka moment, that came in 2003, when he suddenly realized that the unique bits of DNA between the repeats were a perfect match to viruses. He thought, wait a minute, is this a bacterial immune system? But nobody believed him. His paper got rejected over and over. It wasn't until 2005 that his groundbreaking idea finally saw the light of day.
So Mojica figured out what CRISPR was doing. That was huge. But the next giant leap was figuring out how it worked. And, more importantly, how we could hijack that system for ourselves.
All right, so what exactly is CRISPR in nature? The best way to think about it is like a bacterium's personal most wanted list. It's a scrapbook of all the viruses that have tried to attack it before. When a new virus shows up, the bacterium takes its mugshot and saves it for later.
And it's this really clever 4-step process. First, the bacterium grabs a little piece of the virus's DNA. Second, it stores that mugshot in its CRISPR library. Third, it makes a little RNA copy to act as a guide. And finally, and this is the good part, if that virus ever shows up again, the RNA guide leads a protein called Cas9 right to the invader to chop it up. It's simple, elegant, and totally brutal.
Okay, so here's where the story really kicks into high gear. It's 2011 at a microbiology conference. Two scientists who had never met end up having coffee in a little cafe in Puerto Rico. That was Emmanuelle Charpentier and Jennifer Doudna. They realized the research overlapped and decided to team up to figure out exactly how that Cas9 protein worked its magic.
And what they discovered was just mind-blowing. This diagram here kind of shows the gist of it. They figured out that the Cas9 protein is like a pair of molecular scissors, but it's completely blind. It needs a guide. And that guide is a little piece of RNA that basically takes the scissors by the hand and leads them to the exact spot on the DNA that needs to be cut.
But here's where their real genius shined through. They made it simple. See, in nature, the system is a little clunky. It uses two separate RNA molecules to get the job done. Doudna and Charpentier had this brilliant idea. What if we just fuse them together into one? And just like that, they created a single guide RNA. All of a sudden, this complex natural defense system became a simple two-part toolkit, the Cas9 scissors, and a single guide RNA that you could program to cut any DNA sequence you wanted. It was a total game changer.
Once their discovery was published in 2012, the floodgates opened.
Everyone knew this wasn't just some cool academic finding. This was a technology worth billions. And that, of course, set the stage for one of the biggest and messiest legal fights in the history of science, the patent wars.
Almost right after Doudna and Charpentier's paper came out, another brilliant researcher, Feng Zhang, over at the Broad Institute, hit a really critical milestone. He was the first person to show that this system didn't just work in bacteria and test tubes, it actually worked in eukaryotic cells. The more complex cells that make up plants, animals, and, well, us.
And that set off a legal battle that lasted for a decade. It was all about who really invented the tool for use in things like medicine. On one side, you had UC Berkeley, for Doudna and Charpentier, saying their 2012 paper was the fundamental invention. On the other side, you had the Broad Institute, for Zhang, arguing that making it work in human-like cells was the key inventive step. In the end, the Patent Office basically said they were both right, that the claims were distinct, meaning both teams now hold crucial patents on different pieces of the technology.
So, with the legal stuff mostly sorted, the tool was out there, and scientists did not waste a second. So let's talk about how CRISPR is, and I'm not exaggerating, rewriting our world, from the food on our plates to the future of medicine.
Like Jennifer Doudna herself said, one of the first and biggest areas to feel the impact is agriculture. I mean, being able to make tiny, precise changes to crops just got so much easier, so much faster, and so much cheaper because of CRISPR.
And the possibilities here are just staggering. We're talking about simple things like apples that don't turn brown when you slice them. Think of all the food waste that could prevent. But also huge things, like making orange trees resistant to a disease that's wiping out entire groves, or making bananas that can fight off a fungus, or even rice that can grow during a drought. These aren't just tiny tweaks. They could completely change global food security. But of course, the most profound impact of all has to be in medicine. For my entire life, the idea of actually curing a genetic disease by going in and fixing the broken gene, that was just a dream. CRISPR is turning that dream into a reality.
We already talked about the approved therapy for sickle cell disease, which is incredible. But that's just the start. Right now, there are clinical trials happening for hereditary blindness, for muscular dystrophy, for Huntington's. Scientists are even trying to use CRISPR to literally snip the HIV virus right out of a person's cells. The potential here is just, it's hard to even wrap your head around it.
So we've gone on this incredible journey, from a curious observation in a small lab in Spain to a tool that is fundamentally reshaping what's possible. There's really no doubt that this technology is going to help us solve some of the biggest challenges we face. But it also leaves us with maybe the most important question of our generation. Now that we have the power to rewrite the code of life itself, how on earth should we use it?